finish off a little bit of Module 5 and then get stuck into Module 6. And I just did clear it with May that to teachers and students via the teachers, if you wish to reach out to me, tap my brain or exchange ideas, I'd be really look forward to that opportunity to work or exchange ideas with you. So if there's anything that I've covered too quickly in not enough detail, please do take the opportunity to track me down, email me, and I'd look forward to that opportunity to pass on, exchange ideas and information. Okay, and again, just to reiterate, really conscious of the fact we've only really got 1 more hour left to cover what is about 10+ weeks worth of work. So that requires me to speak rather quickly and just jump between some sort of key ideas. So just in terms of, we're getting towards the tail end of Module 5. Just key terms. Look, we cover this in stage 5, so it really is assumed knowledge at this point in time. But just to reiterate, we've got a homologous pair of chromosomes there. For each particular gene, we have 2 copies of that gene. They can be the same or they can be different. So in this particular case at the top there for gene A, we have 2 of the same allele, so an allele being different versions of a gene, and we refer to that as homozygous. Breaking that word down, homo means the same, hetero means opposite or different. And so in this, for gene B there, we've got 2 copies. They are referred to as a heterozygous pair of alleles. Now that will become increasingly important as we work through the different types of inheritance. Whilst it's a bit of a simplification here. iColor is actually determined by more than one gene. It certainly highlights the difference between and the importance of you knowing the difference between homozygous and heterozygous individuals when we look to do the pedigrees and the Punnett squares. So for the homozygous individual and the heterozygous individual, their phenotype, in this case brown eyes, is identical. Okay, so to look at them, there is no observable difference between them, and it is only through looking at pedigrees, Punnett squares, that we can actually determine the likely genotypes, or in some cases the definite genotype of individuals. And the only time at which a individual will express the recessive allele in their phenotype is when they have 2 copies of that recessive allele. Okay, so that's what we call just normal autosomal inheritance. So autosomes being the non-sex chromosomes, which then links nicely through to the next scenario, which is sex linkage. And again, I like to just pause and remind you that we tend to have that human focus on these scenarios for obvious reasons. You should know females XX, males XY. But in the insect, the bird world, there's a whole range of different scenarios where, for example, it's actually the male that might have the same homologous pair of chromosomes, and it's the female that has the differing chromosomes within the sex chromosomes. I tell you that because remember, Band 6 students are able to apply learnt knowledge to unfamiliar contexts. Okay, so we don't go teaching bird reproduction and insects genotypes, obviously. But the knowledge that you take from here when we look at humans is applicable to those other scenarios where it might be flipped on its head and it's actually the female that you're dealing with who is has the different chromosome pairings. Okay. Now, the reason sex linkage, it's important to have an understanding of that, is that with the females there are actually 3 possible genotypes, 2 possible phenotypes. And you represent it with the capital X and then the capital T. I use T for trait for the dominant allele capital, and then the little t recessive allele. And then these 2 individuals, their phenotypes would be the same because they're expressing that dominant allele in their phenotype. Now, because most, not all, but most sex-linked traits are carried on the X chromosome, with males, we have a different scenario as compared to those autosomal inheritance where, because the male only has that 1 X chromosome, they either have the trait or they do not have the trait. Whereas females in this middle example here, the heterozygotes, are what we refer to as carriers. Okay, you cannot have a male carrier, which makes it really important to understand that when we do look at these pedigrees. Quickly jumping through codominance and incomplete dominance, they present they're similar in terms of having an understanding of what they would present as with the pedigrees and Punnett squares. With codominance, it's all in the name really. Both alleles are dominant and will be expressed in the phenotype. The go-to examples always with roan cattle. So there are 3 possible genotypes, and therefore 3 possible phenotypes. And just simply by looking at the phenotype of the animal or the organism, animals here, you can determine the genotype of the organism, where the whites, again, represented by capital letters, capital WW is a white, capital RR red, and then the combination presents with what's referred to as a roan individual, where both proteins or hair colors are actually expressed in the coat color. And what's that look like in a real life example? These are the sort of scenarios that you'd be asked to apply your knowledge to. They're probably easier than the autosomal dominance, to be honest, simply because when you look at the phenotype, you instantly know what that genotype is going to be. And on the right here, just to look at a more interesting scenario, we've got 2 roan individuals, and they can actually result, if you were to apply the Punnett square, capital R, capital W, cross capital R, capital W, you actually end up with 3 phenotypic combinations where we've got 25% red, 25% white, and 50% will be the roans, similar to the parents. Now, the reason I say the codominance is similar to the incomplete dominance, this is relatively new to your syllabus, that we didn't have to teach it in the previous syllabus, that being incomplete dominance. It's similar because there were 3 possible phenotypes, okay? The difference between incomplete and codominance is that incomplete dominance, it is what you might typically think of the merging of the characteristics. So, you know, you ask a year 7 student, what do you get when you mix red and white? They will say pink. That is incomplete dominance, where you're getting the merging of those two phenotypes. Okay, so it's not that both are being expressed equally as with the codominant individuals. It's where you get that blending of characteristics. Now, these will be really easy to pick, incomplete and complete dominance in your pedigrees, because you're going to be looking for, quite typically 3 phenotypes represented by one being coloured in black, one being coloured in white, and then the other might have a cross through it. Okay, so it's an instant sign that we're dealing with incomplete or codominance. Now, the other one I'm just going to draw your attention to is polygenic inheritance. Now, you can forget about it after this discussion if you like. The reason I certainly tap into polygenic inheritance is that if you remember back to the original dot point, it is an including but not limited to, okay? Polygenic, poly means many, and so polygenic inheritance refers to traits that are basically dictated, the phenotype is determined by more than one gene, okay? The best example in all the textbooks is skin color. And in this simplified version here, what we have is 3 genes that are coding for the one trait, that being skin color. And what happens is it's almost like it's a Punnett square on steroids. You have to look at all these different combinations of alleles. And if you have the dominant allele, that means that the effect of that dominant allele is expressed. So 3 dominant alleles expresses each of the different traits, whereas the recessive allele is ignored. So what does that mean? So if the gamete had capital A, capital B, capital C, so 3 dominant alleles, the egg and the sperm each have 3 capitals. What ends up happening is you have 6 dominant alleles, and when you look at that pedigree, it ends up with the 64 combinations, and one of every 64 will have that really dark skin. And then similarly, at the other end of the spectrum, 3 lowercase letters also ends up with 1/64th. And if you were to look at all the various combinations, you get basically a normal curve distribution of those different skin tones. Okay, so it's not being coded for by simply one gene. Okay. Also in this particular outcome is, again, way back at the start of last session, I drew your attention to the fact at a band 6 student really needs to be able to distinguish between those terms, trends, patterns, and relationships. Myself, I'm guilty of just, we put the outcome on and then we start teaching it. But the outcomes themselves really need to be broken down by teachers and students, because if that word appears in the dot point, Nessa has the opportunity or the go ahead, I guess, to assess those words. Okay, with this particular outcome as well, it's for example. Okay, now what that means is I naturally again will teach these frequency data and SNPs, single nucleotide polymorphisms, because they give you the examples. But just to highlight that, if your teachers have used other examples that actually address that outcome, that is okay. Because the likelihood is that it will present as a skills question where you get given the necessary information to answer the question. Okay, so I've talked a lot about trends, patterns, and relationships. So I now make a really active point to define these. Back in junior years, now I start having these discussions. And whilst this particular graph does not relate so much to year 12 content, it does year 11 content, the two graphs really highlight the important differences between a trend, a pattern, and a relationship. Okay, so in this top graph, again, not specific to heredity or mutation, but we're looking at time, and we're looking at CO2 emissions and global temperature change. And what you can see, a trend is the general tendency of data to move in a certain direction. Okay, really important that if asked to identify a trend within the graph, don't just say it's going up. It's not sufficient to get, let's say, if it was a 2-mark question, you would say overall the graph is both CO2 levels and global temperatures are increasing, but engage with data points. In 1964, it was 320. In 2008, it was, goodness me, 380 or thereabouts. So that change in a certain direction, in this case over time, is the trend. A pattern is when the set of data repeats in a predictable fashion. So jumping down to the bottom graph, again, you can see here it is what's across a scale of 400,000 years, but every 50,000 years ago, every 50,000 years, there is a peak, then a trough, peak, trough, peak, trough. It's that repeating cycle of data that's in a predictable way, okay? The relationship is probably more bread and butter, where we're looking at the relationship between the independent and dependent variable. So where, if I could look at an example in this particular graph, quite often we're looking at, I jump back to the top graph, there is a distinct relationship between temperature and CO2 levels. As CO2 levels increase, as CO2 levels in the atmosphere increase, global temperatures increase, okay? There's your relationship. Now, a limitation, often overlooked, but it's important that you look for any holes in the data or that are presented to you. Don't assume that they're giving you all factual information. NESA may require you to actually analyze the data and look for possible, I guess, flaws in it. And as I look at this one here, look how far the time scale goes back. We're going back 400,000 years. Okay, where you and I are up here near zero, so we're relying upon, whilst quite stringent or accurate equipment to measure ice core drilling and so forth to look at these temperature CO2 changes. We in reality cannot ascertain what environmental conditions were necessarily like 400,000 years ago, if that makes sense. So just looking for any holes in the data to address limitations in data. Okay, now I mentioned frequency data. It was for example, but I think this is an important point to make. Okay, students quite often are messing up that link between genes and alleles. Okay, so an allele frequency, so looking at frequency data, that is the fraction of a particular allele within a defined population. Now, just to highlight that with an example, if we have 100 individuals, you have to remember that they are diploid. The majority of their cells are diploid except for the sex cells. What that means that within that population, we actually have 200 copies, because again, you've got a homologous pair of chromosomes, 2 alleles for each gene. They may be the same, homozygous, they may be different, heterozygote. Now, with this scenario, if 40 individuals are heterozygote for allele A, 10 individuals are homozygous for allele A, the capital, the dominant allele. What that then allows us to determine is that we have 40 plus 20 copies of that dominant allele, if that makes sense, coming to 60. If you're wondering where I got that from, it says 40 individuals are heterozygote, so that means they're capital A, little a. So that's where that 40 comes from, okay? 10 individuals are homozygous, So therefore, we need to double that to get the 20 because they've got 2 copies of that dominant allele, okay? And the next, I guess, point to make is that don't make the mistake of then dividing that by 100 individuals. You have to remember we've got 200 copies of the gene in the population. So to work out that the actual frequency of that dominant allele, it is just a matter of dividing 60 into 200, and we're going to get 0.3 in terms of the frequency of that dominant allele. And I always find that really interesting. Students need to appreciate that the recessive allele in this case, there is it's 0.7 or 70% of the gene pool is made up of that recessive allele, and that can be despite the dominant allele being more individuals express the dominant allele in their phenotype, both in the homozygous and the heterozygous form. It can quite often be that the recessive allele is dominant because it's being masked by that dominant allele, okay? And SNPs, in terms of what a SNP is, don't overcomplicate it, I guess. You will have learnt a lot about, in Module 6, point mutations, chromosomal mutations. The difference, in essence, a SNP is a point mutation, but the catch to it is you need to add a little bit more information. It needs to be present in more than 1% of the population, okay? And the reason that is important is that scientists then use that in what I'm going to talk about is genome-wide association studies. And if it's less than 1%, I guess it's not statistically significant to draw, conclusions from in terms of whether that SNP is associated with particular diseases. Okay, so it needs to have been present in a population for a significant amount of time to reach that 1% threshold. Now, yeah, I think it's Alpana. Love the question, actually. So I would say the answer is no. So the question there, if you didn't see it, was do they need to know Hardy-Weinberg? I love Hardy-Weinberg. If I want to extend my kids, is what I'd say is I do touch upon it, but they certainly won't be required to define it, talk about how it's used. What they could be given again is a stimulus with all the relevant information, what P and Q represent within Hardy-Weinberg, and then be able to apply information that is contained within the stimulus. So explicitly, no. Could it be included in a question? I would suggest that yes, it could be, okay? And there's no harm in teaching because it's really interesting and really does sell the message in terms of frequency data and stability of populations and so on. Okay, important point to make with SNPs, what scientists are able to do, and this will link, it does link in nicely with Module 8, where we look at those big studies, epidemiology studies. Just had a mind blank, seniors moment, sorry. But the principle is as follows. We take individuals that might have a particular disease, look, one that a lot of prostate cancer, breast cancer, it can be Crohn's disease, so those irritable bowel syndromes. What scientists do is they take a relatively large, so we're talking thousands of individuals that have the particular condition, and then we have the controls that do not have the disease of interest or the syndrome, whatever it may be. What scientists then are able to do using relatively high-end, technologies we'll explore later, is they analyze genome-wide studies, and they are looking for the presence of SNPs. So these mutations that are present in more than 1% of the population, and whether any of them are actually more prevalent within individuals that have the disease. What that then tells the scientists, the researchers, is that that particular SNP may be associated with that disease that they're studying. Now, important to remember that SNPs can occur in coding and non-coding regions. So it doesn't mean that the SNP is responsible for the disease, but it might be somewhere on a chromosome that is closely associated with a gene that does cause the disease. Hopefully that makes sense, okay? And then some questions. This is one of the questions, or is there more? Just looking at the chat. Okay, yep, back to get rid of the chat. So this is one of the questions I first saw, and I thought, oh, wow, I'm glad it's the students answering it, and it's not me on the spot that had to lock a choice in it. It took me a little thinking about. Hope it would be awesome if some of you jumped in and if you haven't seen this question, give it a go at answering them. answering it. Which diagram correctly models one phase of meiosis in an organism that has 6 chromosomes in its somatic cells? Hopefully you can all discuss amongst yourselves lock an answer in. The thing I'd also add to this, the HSC all papers, the examiners who write the papers or the exam writers, they're under instructions to do as best they can scaffold the paper. Okay, so where question 1, 2, 3 should be relatively easy, and as you get through to your later questions, question 18, 19, 20, the idea is that they're becoming increasingly more complicated. They don't always get that right because we don't see what students necessarily see, but I tell you that because this question 19 would indicate that the exam writers had every intention to make sure students really had an in-depth knowledge of meiosis. Not seeing any chats. No. I'm going to just have to go through it with you. So the answer is, what did I lock in? I've locked in answer A. Okay, and I shall tell you why. Again, go away and think about it and feel free to argue with me afterwards if you think I've got it wrong. I haven't checked the answer, but I'll tell you why. With option B, you can see that this is definitely knocked out because never would you have a scenario where different chromosomes, as in they line up in homologous pairs. Here we've got a long and a short chromosome lining up, so I can instantly knock out option B. With option C, you will note that in the stem of the question, it says that somatic cells are diploid 6. That requires you to have an understanding of the difference between homologous chromosomes and sister chromatids, because this individual here in option C has a diploid number of, I have 24, if my maths is correct. So as a result, we can knock that out because it doesn't address the stem of the question. Now, in terms of what's wrong with option D, it is showing the end of meiosis 2, where we're actually going to get the sister chromatids being separated off. And therefore, what would have happened prior to that would indicate that for this individual, that the diploid number would have been 12, and therefore having that understanding of what's going on in each of these images draws me to the answer being A. Okay. So I'm just going to move forward, again, way behind time, and just look at a couple of pedigrees. Now, before I look at, and this is in my own knowledge as I get my head around, before I go and look at the answer options, there's one particular pattern that I always look for, and that is what I've got circled on this particular image. If you have two parents that have the trait, giving rise to a child that does not have the trait, or vice versa, where you have two parents that don't with a child that does, it instantly tells you whether the trait is recessive or dominant. Okay, so to highlight that, I instantly got my own attention is drawn to this couple over here. We've got two parents that have the trait and a son that does not have the trait. That then tells me that the trait is going to be that both parents must be heterozygotes and that they must pass on the recessive allele to that son. Okay, and the reason I know that is that the only way that these individuals, mum and dad, can have the trait and pass on the recessive allele is that if they have the dominant allele masking the recessive allele. So they must be both capital T, little t, and therefore the trait that we're following in this pedigree is dominant. Okay, so it is therefore, initially it's going to be option A or B. Now, then to determine how it is the difference, whether it's sex linked or not, once you've determined that it is a dominant allele, the carries Australia, therefore is autosomal. Now, also using the same parents, it cannot be sex linked recessive, as the mother would be XT little t, and therefore her son would have been affected, if that makes sense. So the fact that mum is giving up one of her X chromosomes to that son means that the only correct option is going to be answer A. Oops, sorry, there with the squiggles. Now, this question appeared in, there's another one I really was keen to show you, because this one here, 2021 paper. Tricky question, because again, year to year, I teach principles very much teach things this in the same order. And if I'm just rewinding back to that graph on enzyme activity, for many years I said 1 gene, 1 protein, 1 gene, 1 protein. All right, and then I tell students to look for those particular patterns where we've got parents both having the trait and not giving rise to a child with it, and we can determine whether it's recessive or dominant. So initially, when I saw this image, my attention was drawn to these individuals here. 2 parents that don't have it, giving rise to a kid that did. So my instant thought is that the trait is recessive. However, and it's a big however with this particular example, they tell you that it is autosomal dominant in the stem of the question, okay? So the question then becomes, how did this child, this son here, actually end up having the trait, okay? Because it cannot be coming from the parents, because the fact that both parents, if it was dominant, it means that both of these parents must have had the trait, if that makes sense, because this son here needs to have inherited one of those dominant alleles from the parents, and because it's dominant, one of those parents needed to have the condition in question, okay? And so here is this trait likely to be, I'll just jump over to the marking criteria here. You needed to, again, looking at the actual stem of the question and how that marries up with the marking criteria, again, demonstrating the importance of you analysing the question in depth before committing pen to paper. So is this likely to be the result of? So you need to identify what it is, and look here, for 3 marks you needed to identify the type of mutation. The question required you to justify, so you are absolutely guaranteed that the marking criteria will be linked to your justification, and sure enough, there it is. So it is a somatic mutation, so that is occurring within that son, okay? So it didn't occur, it wasn't present in the parents. And the reason for that, your justification is going to be, as I said, that it's not present in either parent. And because it's a dominant trait, it tells you that in the stem of the question, it would mean that one of those parents needed to have that particular trait. Okay. So, going to skip a few of the pedigrees that I had so I can get on to Module 6 shortly. Note that the booklets that went out to you did have a lot of extras with the marking criteria in them. So, yeah, please enjoy yourself. Is enjoy the right word? Tackling those after we finish, okay? So just moving on to, well, for example, again, it's another outcome that has for example, but look, I'd strongly urge you to go with DNA sequencing and profile. Reasons will become obvious when I look at some of the questions that they use. Again, all the information is typically within the stem of the question. And this is an investigation which requires, perhaps with teacher-led discussions, how different technologies are used to determine inheritance patterns. Now, the obvious ones when we look at DNA, oh, that's a bit messy, DNA sequencing and profiling is gel electrophoresis and PCR. So just to take you through those, okay. I'll just jump through to here. There's 3 key stages involved in PCR. We need to denature. So the principle behind PCR is that you typically end up with a small amount of DNA from a sample, and we need to produce lots of copies of it, okay? And the way that we produce lots of copies of it is through PCR, okay? And the principle is as follows. We have a segment of DNA, the double helix. The first thing that you need to do to make copies of it is to unwind it, okay? And that's what we refer to as denaturing, okay? And so denaturing, if you think back to those year 11 pracs you did where you looked at enzyme activity, effect of temperature, denaturing is just where you're basically disrupting the molecule. And what it does is the temperature of around 95 degrees Celsius separates the hydrogen bonds or breaks the hydrogen bonds between each of the backbones. What then happens? The process of annealing is used. And within the PCR tubes, you've got lots of these, the free-floating nucleotides, your guanine, cytosine, adenine, and thymines. They come racing in and they bind to those now two exposed backbones. And then you allow that to occur for a period of time. Temperatures reduce for around 2 minutes, and we get elongation of that, now the two backbones of the original DNA. Now, if you repeat that over, it's typically around 30 cycles, can be less, can be more, but what that does is produce a really large amount of DNA with the scientists to work with, okay? But at this point in time, it's important to note that PCR in itself is only about amplifying the DNA. You then need to do something with it, which is where we start looking at DNA profiling and DNA sequencing. Now, just quickly, the DNA, the main technique used there is gel electrophoresis. As the name suggests, well, it's a gel, it's an agarose gel, and the samples of DNA are placed within little wells at the top, which is, I guess, image 3 here. We put our samples in wells, a current is applied. Those DNA fragments, they actually have small charges on them. They can be of different sizes. The smaller charged particles, common sense approach, will actually migrate more quickly through the gel. The larger particles, they're more cumbersome, find it harder to move through the gel. And then what we end up with is a sequence of gels that can be used. In this case, we're looking at DNA profiling, often used in either crime scenes or paternity tests. And this is the sort of thing that you will get in the exams, okay? You won't be asked to define necessarily gel electrophoresis or PCR. You get given this stimulus and asked to interpret the information within it. And in this case, we've got obviously the mother is known, the child is known, and we've got two prospective fathers. What you then need to do is look at the DNA fragments in the child's sequence, or profile, I should say. You look up the bands or cross-reference the bands between mother and child, because we know that those particular segments have come from the mother. And what that then means, in keeping in mind you get half your DNA from mum, half from dad, the other bands need to will have come from the father. Okay, so as soon as you see these in highlighted here, bluey purple color, that means that male 1 is the likely father of this child, because those other DNA sequences found in the child are not found over here on male 2. Okay. All right. Now, arguably the biggest outcome in the module, but the good news is it is a for example. Okay. Now, I would strongly urge, you can go to town and do all three. You could be doing group works where one group did conservation management, the other inheritance of diseases, human evolution. There's real scope for some really interesting studies, or even depth studies. But in the day-to-day teaching, if my delivery of it deals specifically with one of them and one of them only, and that being the inheritance of a disease or disorder. The reason I say that is that all of module 7 and 8, we're dealing with infectious diseases and non-infectious diseases disorders in module 8. Why reinvent the wheel when the big mark questions in the HSC now tend to be crossover between all different modules, okay? The fact that it's a for example means that you don't have to do all three of them. You can choose one or do a totally different example of your choosing. I go with that middle one there because of the links, obvious links it has with modules 7 and 8. The other thing I'd say is note in your responses, this is all about large scale collaboration projects. And here we have again that focus that I'm trying to emphasize on trends, patterns, and relationships. Okay, so you really need to know what those terms mean and how to apply them to a given scenario. The big one for me is as a starting point, it doesn't get much more of a large collaboration than the Human Genome Project. It involved, oh goodness me, I'm going to say approximately 20 labs around the planet or around the globe. Started back in 2003, and what that came up with are these collaborations. Different labs were in charge of sequencing different chromosomes. It determined that we had 20 to 25,000 genes with about 3 billion base pairs. And the follow on from that is that from these large collaboration projects is that we they form the basis of these studies that are now being performed in a collaborative nature, looking at the causation of specific diseases, if that makes sense, because we now have the full sequence of the human genome. That then allows us to look for those, if you remember back to the human gene, the genome-wide association studies. Here, I just got an image of that here. This is the flow on from that, where we can now go and explore regions of DNA and look for links to specific diseases, okay? Nearly into module 6, okay? Just going to focus on this one particular one, okay? And just to highlight 2021 paper. Again, no, it's all in the stimulus. Everything you need to know here is in this stimulus. Okay, it is essential. Pen down, highlight the question, and look for those key words. Okay, a part of the DNA profile is shown, and it shows the alleles present. Okay, so step one, you need to understand that mum and dad are both heterozygotes. They tell you that both children, child 1 and child 2, are free from the condition. And so what that means is that this individual is homozygous dominant, and this individual is heterozygous again. And the reason that they don't have the disease is that this dominant allele here is masking that top recessive allele. What that then tells you is that child 3 must be homozygous recessive, and then using the DNA profile justify whether child 3 will have sickle cell anemia. One mark, if we jump forward to the marking criteria, it's going to be identifying that the child 3 will have sickle cell. There it is there, child 3 will have sickle cell. And then that justification is for those reasons that I've just given you. So you can see that again, all the information you need is in the stem of the question, but what you really do need is that in-depth knowledge of genes versus alleles, heterozygote versus homozygous, and be able to apply them, okay? And again, to bore you to tears with the same old message here, some relevant information. So if you've got no idea what's going on with this particular question, but if you start putting down the terms homozygous, heterozygous, okay, you can't give conflicting information, but if the information is relevant and not in the stem of the question, you're going to get a mark, okay? Okay, interest of time, I'm going to now jump right over to module 6, which is genetic change. Firstly, how does mutation introduce new alleles into the population? Okay, just to reiterate, a lot of students think that mutations are bad. Okay, important that you understand that is not the case. They certainly can be, but you need to appreciate the role that mutations apply in the variation within a species and also survival of that species and long-term speciation. Okay, you and I tell my own students we'd still be bacteria swimming in the, I think the current knowledge is mid-oceanic ridges if it was not for mutations and the introduction of new alleles. Okay, the dreaded including but not limited to, you need to know examples of EM radiation chemicals and naturally occurring mutagens. What's common to all of them is that they lead to a change in the genetic sequence. By definition, that's what a mutagen is. A mutagen causes a mutation. They can do that in different ways. Okay, then jumping through to this one. Again, I can't go over all of them in depth, but what can happen here is within the electromagnetic spectrum, the one of particular focus here is the ionizing radiation. Okay. So you do the EM spectrum back in stage 5. The ones of interest here are the ones that have a really short wavelength. Yeah, just had to get my junior science right there. This what's referred to as ionizing radiation, e.g. UV, X-rays, gamma radiation. They're able to interfere with the bonding between the nitrogen bases, okay? And that can then have various flow-on effects. It can be a point mutation. It can be multiple base mutations that occur. But at its core, the ionizing radiation disrupts the structure of DNA and can insert, delete nitrogen bases from that sequence of DNA, okay? Why is that important or relevant? Again, linking this now back to module 5, that's where the big mark questions come from. Understand how that links into this process here, okay? Protein or polypeptide synthesis, transcription, translation. Again, I have seen questions that require you to understand that a mutation occurs at the DNA level. If a base changes in the mRNA or the tRNA, it's not really going to have any major significant effects because it stops with that particular change, okay? Whereas if it occurs back in the DNA, that can be passed on and have an impact on that resulting protein. Now, engaging with your your amino acid wheel, just to highlight that here, that if you were to follow transcription translation, that if you had a U, a C on your mRNA, it would produce the tyrosine amino acid. Now, a simple change that changed that third, that cytosine, say, to an adenine, you would get a UAA. and that ends up in a stop codon. Okay, that can be absolutely catastrophic because it stops the production of the relevant protein in its tracks. Okay, and therefore you'll have a non-functional protein. The flip of that, however, is, in contrast to that is, you can appreciate that if you had a UCU and at that back at the DNA, if a mutation occurred that led through to a mRNA sequence that went from a UCU to a UCC, it produces the same amino acid. And as a result of that, the impact is going to be negligible, not necessarily no impact, but it can have minor effects on the overall, like the secondary structure. But compared to a stop codon insertion, it's nowhere near as significant. Okay. Now, another interesting one, because it's including but not limited to, you'll note it says point mutation, chromosome mutation. My own teachings, I look at all the different types of points and chromosomal mutations, but you won't be asked to define a nonsense mutation or an insertion mutation, okay? Because it's not defined in the dot point. You can therefore not be assessed explicitly on the word that is not included in the actual dot point. They can give you scenarios, and you just need to understand how that flows through from DNA down through to the actual final product of a protein product. So taking you through them really quickly here, we've got the normal DNA there. We've got the, what's that? That's okay. It's the template strand, okay, as compared, contrasted to the coding strand. And we produce this mRNA sequence, those 4 amino acids there. What we've got is a point mutation. It's all in the name, a point. It's a single point. We get the mutation where the adenine is converted to a guanine, and the flow-on effect of that is that we have a change in a single amino acid. Okay. Oh, sorry. In this case, the amino acid is the same. Okay, phenylalanine there. Now, that is what we refer to as a synonymous mutation. Synonymous, just in general usage, is the same, and so it's likely to have very minimal impact on the resultant protein. Now, non-synonymous are broken into missense and nonsense. Look, I like to say missense, a bit of mischief, whereas a nonsense, it's absolute nonsense, the product, okay? And so with a missense, it's where we do get a single change in an amino acid, which can change the secondary tertiary structure of the protein, which can absolutely impact its function. Whereas a nonsense mutation is that catastrophic one where we get a stop codon being inserted, which is just produces a non-functional protein. And then finally, frame shift deletions or insertions over here. The frame shift mutation, think movies, the old school movies where you have frames in a movie, and what happens is that by the insertion or the deletion of a nitrogen base on the DNA, everything gets moved up, okay? That typically, not always, but typically is catastrophic again, because whilst it doesn't insert a stop codon, what it does is change nearly all the amino acids that follow that particular mutation, and therefore changes the sequence, changes the structure, changes the function of the resultant protein. Okay, so chromosomal mutations, probably the main one we look at is the bottom one, and you'll be familiar, I anticipate, with trisomy 21, commonly referred to as Down syndrome. Okay, you need to know that there are other types of chromosomal mutations. Remember, the outcome was an including but not limited to. So you have to have some sort of sense of the different types. Again, I can't reiterate it enough. You're not going to be asked to define each of these terms, but you would need to be able to apply and understand what the consequences of those mutations would be. Okay, so with the non-disjunction trisomy 21, it's where during meiosis we don't get the correct splitting of sister chromatids and or homologous chromosomes, depending upon where it happens, and we end up with a daughter cell that has 47 chromosomes rather than the typical 46. Okay. So again, next two dot points, I've grouped them for, I think for obvious reasons. You need to know the difference between those somatic germline mutations. It's concerning that some students at this stage don't know what somatic means. It's all your diploid cells. It's all cells other than your gametes or the germline cells that actually give rise to those gametes. So you need to know the difference between and the significance of a mutation occurring in your somatic cells versus one occurring in the gametes, oh dearie me, messing up the underlining, or versus those germline mutations. Now, that then leads nicely onto that second dot point, assess. You need to make a value judgment, okay? Don't just identify and give an overview with a description or an outline. You need to commit to that value judgment because it's an assess, and understand the impact of a mutation that occurs in the coding and the non-coding regions in eukaryotic organisms, okay? And I would hope that you can appreciate that if a mutation occurs, we've just looked at it, if it occurs in the coding region, my value judgment would be that it can be catastrophic and therefore have much more significant impact on the resultant gamete or organism. We have those non-coding regions that sit outside the genes, but also within the genes between the coding regions. If a mutation occurs in one of those non-coding regions, it tends to have less impact or borderline no impact on the code on the organism, because those non-coding regions, they can actually influence the coding regions if they occur quite close to the coding regions. However, if they're outside of the genes of interest, they typically have less effect, but you also need to know that they can influence the expression of the coding regions. Hopefully that makes some sense. And nothing beats a good image. This just highlights the point that a germline mutation, so it's occurring back here at the egg or sperm, every cell in the resultant offspring will express that mutation, and therefore from fetus right through to adult form, the cells carry that recessive allele, and depending upon whether you're homozygous or heterozygote, you'll note down here that it's quite not always in this case the male, it's the sperm there, they may carry that the allele in question or the mutation, or they may not. Okay, that's simply because of that, through meiosis we get that haploid number of chromosomes in the resultant gametes. Now, in contrast, somatic mutations can be obviously catastrophic for the individual. Think skin cancer, EM spectrum, that causes a mutation in a skin cell and gives rise to skin cancer. That basically stops with the individual. Okay, so it can be very problematic for that individual, but because it is not carried in every cell of the individual, what happens is when the egg and sperm, in this case sperm, are produced, they will not carry that mutation. Okay, so it stops with the individual in question. Let's have a quick look at time, 12:04. Oh dear. So I'm going to pick out some of the good stuff now. Okay. This is just the outcome here. Investigate the cause of genetic variation relating to the processes of fertilization, mutation, and mutation. Okay, so you really need to be well briefed in understanding that all variation, its original source is via mutations. Okay, they then get passed, or I guess proliferate through the individual, and upon the formation of their gametes, fertilization can then pass on those mutations. So fertilization is certainly important. But the big focus I would suggest is you really need to have an in-depth understanding of meiosis and how that variation is generated through crossing over, through independent assortment, and through random segregation, which then leads me to this particular image. And just to highlight that we've got two possible outcomes here. So we've got a parent cell that is diploid 4. And after we go through, this is going through meiosis, what can happen is that because of independent assortment, we've got, and again, it's chance, we've got the two blue chromosomes on the left-hand side and the two reds on the right. Now, as a result of that, and just to hit rewind, it's important that you understand that homologous chromosomes are the blue and the red one at the top, and the sister chromatids are the two blue ones at the top, if that, hopefully that makes sense. Quite often students think that the two blue ones are the homologous chromosomes and the two red ones are. That is not the case. You get one set from mum, one from dad, and they are the homologous chromosomes. So similarly, these two at the bottom here are homologous chromosomes, and because they've duplicated, the two blue strands here are the sister chromatids. Now with option 1 here, we end up with this combination of the long blue, short blue in the resulting combination of alleles, and then similarly via this, I guess, the product of meiosis 1, we can actually end up with that combination of long red, short red. Now compare that and appreciate that here we have the same starting point, and depending upon how those chromosomes line up along the metaphase plate, you can see that we've ended up with a different scenario after meiosis 1, where we've got the long blue and the short red, long red, short blue on the right-hand side. And then the result of that is we get a different combination of chromosomes or alleles in the resulting in the resulting gametes that are formed. And now imagine the number of possible combinations. We're looking at a parent cell here with 4 chromosomes. We've got 23 pairs of them, okay? So this is a gross oversimplification of what actually happens, but just looking at one particular combination, appreciating the huge amount of variation that can result. And we have not taken into consideration in any great detail crossing over here as well, which occurs between sister chromatids of opposite homologous chromosomes. Hopefully that makes sense, okay? Now, these are terms that are mutation. Students are typically quite comfortable with that. But where, again, there's a lot of confusion around gene flow, genetic drift, and gene pool. Okay, so first step is certainly ensuring that you know the difference between them, where gene pool is the total genetic variation within a population. The gene flow is, as hopefully as the name suggests, is that movement of alleles between populations, okay? And it's very much influences, I guess, evolution, speciation, because for that to occur, gene flow needs to be, I guess, negligible, non-existent, because if there's gene flow occurring between populations, it means that there is that exchange of genetic material. Now, genetic drift, there are typically 2 big examples we look at, and that is the bottleneck effect and founder effect. I would suggest it is incredibly important that you understand the difference between genetic drift and natural selection, okay? Genetic drift is random, it occurs by chance, okay? And then the 2 big examples to highlight that are looking at the founder effect and the bottleneck effect. And so on the left here, the founder effect, it's not too hard to imagine. Darwin's finches are the take-home example. We've got a really big population back on mainland South America, and some of those are going on their normal migratory patterns, but they might get isolated, strong winds, rainfall, whatever it may be, and a small population gets isolated from that parent population. It is chance, it's random as to whether the gene pool found in this small population is the same as that parent population. And once that small population, migrating population, has become isolated, natural selection will then work upon them. And what happens in this case, the red ones are obviously not particularly well suited to the new environment. They get selected against, and we end up with a population that is different to that parent population. Now, similarly, the bottleneck effect, it's all in the name. We've got a original gene pool. What happens is that events occur. It could be due to geographical isolation, overhunting, cheetah population overhunting, climate change, whatever it may be. What then happens? Yep. Sorry, was that you, Marnie? Sorry, jumping in. No, okay. So what then happens is that when the resulting populations, surviving populations, can be distinctly different from that original population, okay? And then the new population becomes distinctly different, and therefore we can end up with speciation that occurs. But again, it is a random event or chance event that occurs, and once that event occurs, that can then, natural selection can go about its business and select individuals with those favourable traits. Again, look, just to highlight how these questions will typically come up. Again, SNPs, you don't necessarily need to have known too much about SNPs except to, because it's all in the stimulus, okay? About 10 million SNPs found in the human genome. We've got 4 modelled here. Which is the best explanation for this? And the answer is because the SNPs model do not affect the phenotype of the individual shown, the correct answer is going to be B, that the SNPs are not found in DNA, so they're in the non-coding regions of the DNA, okay? I only have about 4 minutes left with you. What I'm going to do is go right to the very end. I do apologise. There's just so much good content to get through. There was just a question that I'm quite familiar with. I don't think I can go basic with the marking of this particular question. It was last year's paper. It was question 33, 15 marks. Okay, this is all about the use of these reproductive technologies. A lot of the markers went scratching their heads afterwards and had to again rethink how we actually teach the question, and I'll tell you why with part C. There it is there. Okay, so part C of that question, it's sort of a standalone question. Transgenic species can reproduce and pass on the dominant transgene, okay? Reproduction for aquaculture is strictly controlled using a variety of techniques in order to protect and preserve biodiversity. Now, the major pitfall for this question was, and students answering it and us teaching it, was that it really required students to have quite a complex understanding of the 3 levels of biodiversity. Again, I was guilty of it in my own teaching. I tend to focus on ecosystem biodiversity. But what this question required you to do was engage with genetic biodiversity, so the variation within the population of transgenic versus wild type fish, but also within the species / ecosystem, where what might happen if this the transgenic fish escaped into the wild. Now, in order to get this was worth 9 marks, so it's a significant, a lot of marks allocated towards it. And I just, again, knowing about this firsthand, you simply needed to engage with each of the 5 points and understand how they protected, and here it is, really important, the and preserve biodiversity. So you needed to understand the difference between protecting and preserving the biodiversity. And I'm thinking time-wise, I'm going to finish on this next slide. Again, just looking at the breakdown of that. You needed to engage with the biotechnology for 9 marks and look at how it affected reproduction and biodiversity. You needed to analyse all the different techniques and in specifically pretty much all 5 of those techniques that were mentioned in the question, and importantly how they protected and preserved biodiversity. Okay, so back here to 1 and 5, they were the most easily accessed, because it's the one students are most familiar with, because what this is doing is it's about keeping those transgenic species in an environment that secludes them or prevents them, quarantining them in short, that actually protects that species and ecosystem biodiversity, because we don't want that transgenic species escaping into the wild and impacting the ecosystem. The other 3 through the middle are a little more complex. And you needed to appreciate I just want to draw your attention to one of them, which was technique 3. The sperm produced is collected and used to fertilize egg obtained from wild type non-transgenic species. And where most marks were lost is students not understanding why that was done. And that was done to actually maintain genetic biodiversity within the population of transgenic species. Okay, and if you were to go back and look at the marking criteria, if you're not accessing that particular point, you actually end up down in the 5 and 6 mark bracket at best. So I have missed a lot there of good stuff. And again, I just reiterate, please, if students, teachers wish to reach out to me, I love engaging with students and teachers that are engaged and want to learn and exchange ideas. So I'd be more than happy to extend that offer to everyone.
Thank you so much, Tim. Thank you everyone for your attendance. I hope, is that a legitimate question in the chat? Do students need to know Hardy Weinberg?
It is. It's a very good question. I answered that in just in passing. The answer explicitly no, but it could be given in a stimulus and with the relevant information that they would then plug into Hardy Weinberg would be my strong suggestion. Yeah.
Okay, fantastic. Thank you so much. Thank you, everyone. Our next session is from Dr. Sylvia. She is presenting via recording, which I've just shared into the general channel. So if you wanted to go and watch that recording, and then we'll be back here this afternoon with Halima Haywood, who will be presenting on non-infectious disease and disorders and the exam approaches for that. Thank you so much, Tim. Thank you, everyone.
Thank you all the best.